Assalamualaikum and good day. I'm Binti Ahmad Ghazali, and I would like to present about my final year project report, which entitled The Identification of Bacillus Laculiformis from Soil Using Culture-Based Method and Bioinformative Tools for CCP Aging. And I'm under the supervision of Dr. Razana Binti Muhammad Hanafi. As for the introduction, I will start with the background of study. Bacillus laculiformis is a gram-positive rod-shaped bacteria and endosperming bacteria. This Bacillus laculiformis has the potential to degrade plastic. We have isolated this bacteria, which is Bacillus laculiformis, from mangrove and commercialized soil, and we have identified this bacteria using culture-based method and bioinformative tools. Problem statement. Conventional culture-based method need longer time to identify bacteria, within 2 to 5 days. The difficulty for species identification also due to the closely related species that show similar colony morphology and biochemical reaction. Besides that, the increasing plastic production with a slow decomposition rate and inappropriate disposal might result in a build-up of plastic in the marine environment, which would be difficult to remove in the future. Other than that, the lack of biodecomposable bacteria is one of the reasons why we need to isolate these biodegradable bacteria, such as Bacillus laculiformis. Thus, it will be one of the safe ways to degrade and reduce plastic accumulation. Moreover, the usage of hazardous chemicals that used to compose the plastic also might cause harm to the environment. There are 5 recent questions in this study, which are how to isolate bacteria from the soil. The second one is how to design a specific primer for the gene of bacteria. The third one is how to analyze the specific gene of bacteria. The fourth one is what are the criteria for a good primer. And the last one is how to determine the specificity of primer. Next is significance of the study. The microbe identification by microgen identification kit is capable of identifying unknown bacterial strain specifically rather than using the conventional culture method. The identification of specific genes of Bacillus laculiformis was approached using bioinformative tools, and these bioinformative tools help to obtain the specific primers for gene CCPA of Bacillus laculiformis. Bioinformative tools also useful to further study in molecular such as Pcr, and the isolation of Bacillus laculiformis will help in further study as the biodecomposable bacteria for plastic. Objective of study: to isolate Bacillus laculiformis from soil using biochemical tests. The second one is to design specific primers for CCPA gene of Bacillus laculiformis, and the last one is to analyze the specific gene of Bacillus laculiformis using bioinformative tools. For the scope of study, this study aims to identify Bacillus laculiformis from soil and analyze the specific gene, CCPA gene, using various bioinformative tools. And these bioinformative tools help in achieving good data for dry lab in gene study. Next, for the limitation of study, not enough time to cover on wet lab PCR and further testing on bacteria as biodecomposer of plastic. Next is the literature review. Bacillus laculiformis is a gram-positive bacteria that abundant in the environment such as soil. This is due to its potential as a decomposer bacteria. There are 9,692 genomes of Bacillus species that have been reported in the NCBI. The research about gene expression of Bacillus species show that Bacillus is a helpful alternative in producing various enzymes and chemicals. For example, the CCPA gene, which is the specific gene of Bacillus laculiformis, is involved in regulating biofilm formation and spore production of Bacillus laculiformis. Research by Santosh Kumar in 2013 stated that plastic is difficult to degrade due to its xenobiotic origin that make them endure in the environment for a long time. Bacteria are able to synthesize enzymes to convert plastic polymer to microbial biomass and gases. For example, Bacillus laculiformis can synthesize biosurfactant to degrade commercial polyethylene. Then, the molecular-based method is a tool to analyze microorganisms in soil, and the widespread usage for the bacterial genotyping have resulted in more sensitive results. Bioinformatics is a new scientific field used in information technology to organize and distribute biological data to answer the complicated biological issue. The designing primer is the important step in PCR. There are several bioinformatic tools that are available to select primer pair from a template sequence. Next, for the methodology, we divide it into two parts, which are culture-based method and molecular-based method. For the culture-based method, we start with the soil sample collection and isolation of bacteria. 5 mangrove soil samples have been collected from Kuala Perlis, and 1 commercialized soil sample was obtained from the shop. Next, we proceed with the tenfold serodilation until the dilution of 10 to the power of -9 to estimate the concentration of bacteria in soil sample. Next, we proceed with spread plate method to isolate individual colonies from a dilute and mixed population of microorganisms. Then, we proceed with the bacterial screening. The isolated individual colony was continued with morphology and microscopic analysis of bacillus species using gram staining method and view under a microscope with 1000 times magnification. After that, we proceed with biochemical test of bacteria using catalase test and microgen bacillus ID kit. For the molecular-based method, we started with DNA extraction using real genomic DNA extraction kit. Then, we proceed with the genomic visualization using agar gel electrophoresis and UV gel imager. And then, we continue the method with gene and primate study using bioinformative tools, which are BLAST, FASTA, Cluster W, and in silico PCR amplification. The next part is result and discussion. Tenfold serodilation method was used to estimate the concentration of bacteria present in the sample. From the result, in table 4.1, we can say that the concentration of bacteria in mangrove soil was higher than in commercialized soil. This is due to the characteristic of mangrove soil that can support the growth of microorganisms. Thus, we can conclude that the higher the dilution, the lower the concentration of the bacteria appear on Newton agar from spread plate method. Spread plate method was used to isolate individual colonies from a dilute and mixed population of microorganisms. For the gram staining result, the colony morphology of mangrove soil sample 1, 2, and commercialized soil show the characteristic of bacillus species, which are gram-positive, rod-shaped, and appear purple color. Another type of isolate bacteria was gram-negative bacteria with cocker shape and appear pink, which show the characteristic of other species of bacteria. Further identification of bacteria was proceeded with biochemical tests, which are catalase test and microgen identification kit. For the catalase test, it has shown positive reaction with the presence of bubbles. Next, for the microgen bacillus ID kit, after 48 hours of incubation at 30 degrees Celsius, the color of the well have changed. And there are some advantages of using microgen bacillus ID kit, which are it is a simple and reliable bacterial identification system for environmental sample. It shortens time, reduces cost, and enhances accuracy of identification of unknown bacteria isolate. There are 24 biochemical tests in microgen bacillus ID kit. After 48 hours of incubation at 30 degrees Celsius, the biochemical test that shows positive reaction was recorded to obtain the octal code. This octal code will be entered in the microgen ID software to identify the specific bacillus species. For the mangrove soil sample 1, the bacteria isolate was identified as Bacillus licheniformis with the highest percent probability, which is 99.41%. Next, for the bacteria isolate from commercialized soil sample, it was also identified as Bacillus licheniformis with percent probability of 89.97%. For the molecular-based method, we have successfully extract the genomic DNA of 5 Bacillus species. And from the figure 4.13, it shows the DNA extract of Bacillus licheniformis from mangrove soil sample 1. And the other Bacillus species that we have extract the DNA are Bacillus subtilis group, Bacillus licheniformis, and Bacillus macerans. For the genomic visualization on agar gel electrophoresis, it can be seen that the band has successfully migrated at length 3 and 4, and size of the band is 5,000 base pair. However, the band was not appeared at length 1, 2, and 5. This due to the problem during DNA extraction and skill performed during gel loading. Bioinformatic tools were used for designing primer of CCPA gene. BLAST was used to compare the genomic sequence with high similarity of other organisms and to know about whole genome of organism. Our target organism is Bacillus licheniformis. From the result, in the yellow box, it shows the 100% query cover for 100% identical in most Bacillus licheniformis organism. And on the BLAST result, it also shown 100% of similar organisms that have been selected. Query cover is the percentage of query length included in the alignment segment. Based on the result, it shows 100% for query cover, meaning that it was fully matched to the alignment segment. Next, for the percent identity, it also shows 100%, meaning that the alignment was fully accurate. FASTA or FASTA is another sequence alignment tool that searches for similarities between DNA sequence. Based on the figure 4.16, it shows the gene sequence for CCPA gene of Bacillus licheniformis strain TAB7, and the size of this gene was 4,367,367 base pair. Within the genome, it has the unique region for forward and reverse primer sequence. So we can design our forward and reverse primer based on this sequence. Cluster W is a multiple sequencing alignment program for searching the gene's unique region based on the alignment sequence. In this study, we use Cluster W to align all nucleotide sequence of different Bacillus species that we have obtained from the BLAST end, which have the similar nucleotide sequence with Bacillus licheniformis. From this, we could view the unique region of CCPA gene of Bacillus licheniformis. And at the nucleotide number 231 until 247, we have obtained the sequence for forward primer. For the sequence of reverse primer, we have obtained the sequence at the alignment number 424 until 438. And for the reverse primer, we have to convert it to the complementary bases, which is for G, we have to convert it to C, and from A, we have to convert it to T. These are the forward and reverse primer for CCPA gene of Bacillus licheniformis that we have designed, and this pair of primer have met several parameters, which are the GC content of the nucleotide sequence of primer was 60%, the primer length was 16 base pair, and the melting temperature of the primer was 60 degrees Celsius. The last bioinformative tools that we use is in silico Pcr Amplification. It is a software that helps us to identify whether our design primer is ready to be used for real web lab PCR. And this figure 4.18 shows the PCR product for Bacillus licheniformis in FASTA format. We can detect 3 different strains of Bacillus licheniformis, which are Bacillus licheniformis 9945A, Bacillus licheniformis ATCC 14580, and Bacillus licheniformis DSM13. And from the result also, we can know the size of the amplicon, which are 207 base pair. And from this result, we know that our design forward and reverse primer able to detect target species specifically for CCPA gene of Bacillus licheniformis. In conclusion, the conventional culture method to identify Bacillus licheniformis using a microbe identification kit was successful with a percentage probability of 99.41% for mangrove soil, whereas for commercialized soil, it was 89.97% after 48 hours of incubation at 30 degrees Celsius. Bioinformative tools was a computer software that aids the primary design process for the specific gene of Bacillus licheniformis. A pair of primers have been designed according to several parameters, which are GC content was 60%, the primer length was 16, and the melting temperature was 60 degrees Celsius. The specificity of the primer and the analysis of the gene were done using in silico Pcr amplification, and it showed that the primer were able to detect Bacillus licheniformis strain with the expected size of Pcr amplicon, which was 207 base pair. For the recommendation, in future work, it was an urgent need for researchers to fully utilize the potential of bacteria as decomposers because it was valuable for plastic waste treatment. The identification of bacteria can be done using molecular-based method, as it is a rapid way compared to culture-based method that needs more time and lab work. Few methods and techniques for future study, such as wet lab in DNA amplification and multiplex PCR, as both of these methods can detect multiple genes in one sample, and also further up to sequencing for the specific gene of bacteria with diverse potential. And the last recommendation is the isolation of Bacillus licheniformis also can be used to test biodegradability in plastic.